Over 85% of the world's population possesses a fascinating biological marker, Rh, positive blood. This indicates the presence of a specific protein in their red blood cells, a protein also found in rhesus monkeys. This seemingly insignificant detail has far-reaching implications, shaping our understanding of genetics and human ancestry. However, a smaller portion of the population, roughly 15%, lacks this protein, earning them the designation of Rh negative. This minority group, with their unique blood composition, has become a source of endless fascination and debate, blurring the lines between scientific inquiry and age-old myths. Our blood, the life force coursing through our veins, holds secrets yet to be fully understood. The very essence of our being, it carries whispers of our past, clues to our present, and perhaps even hints of our future. The existence of Rh negative blood challenges our understanding of human evolution, prompting us to question the very foundations of our origins. What does it mean to lack this seemingly ubiquitous protein? Is it a simple quirk of genetics, a random mutation, or could it be something more profound, a legacy passed down from a time before recorded history? The answers, like the blood itself, are complex and often contradictory. Scientists grapple with unraveling the genetic puzzle, while historians and anthropologists delve into ancient texts and cultural beliefs, seeking answers in the echoes of the past. The journey into the enigma of Rh negative blood is a captivating one, filled with twists and turns, leading us down paths that challenge our preconceived notions and force us to confront the unknown. The more we learn, the more we realize how much remains hidden, a testament to the enduring mystery of our own existence. The science behind blood types is intricate, involving a complex interplay of genes and proteins. For the majority of the population, the presence of the Rh factor is a given, a seemingly insignificant detail inherited from their ancestors. But for those with Rh negative blood, the absence of this protein raises more questions than it answers. How did this variation arise? Was it a random mutation, a spontaneous change in the genetic code, or could it be something more deliberate, a sign of ancient genetic manipulation? Theories abound, each more intriguing than the last. Some point to the relatively recent emergence of the Rh negative blood type, speculating that it arose within the last few thousand years. This timeline coincides with the rise of early civilizations, leading some to propose a link between the two. Could the Rh negative blood type be the result of advanced genetic engineering by a long-lost civilization? Or is it simply a natural variation that arose by chance and spread through specific populations through migration and intermingling? The search for answers takes us deep into the annals of human history, examining ancient texts, analyzing archaeological findings, and tracing migration patterns across continents. We find ourselves drawn to isolated populations with unusually high concentrations of Rh negative blood, such as the Basque people of Spain and France and the Eastern Oriental Jewish communities. Could these groups hold the key to unraveling the mystery of Rh negative blood? Do their unique genetic histories offer a glimpse into the origins of this intriguing blood type? The exploration of Rh negative blood is as much about what we don't know as what we do. It forces us to confront the limits of our understanding, to acknowledge that our knowledge of genetics and human evolution is far from complete. It challenges us to consider possibilities that lie beyond the realm of conventional science, to entertain the idea that our past may be far more complex and mysterious than we ever imagined. The fascination with Rh negative blood extends beyond the scientific community, capturing the imaginations of writers, artists, and conspiracy theorists alike. It has become a staple of popular culture, appearing in books, movies, and television shows, often shrouded in an aura of mystery and intrigue. From tales of ancient aliens to theories about secret societies, Rh negative blood has become a symbol of the unknown, a blank canvas upon which we project our fears and fascinations. As we delve deeper into this enigmatic subject, we must tread carefully, separating fact from fiction, evidence from speculation. The journey is fraught with challenges, but the potential rewards are immense. By unraveling the mysteries of Rh negative blood, We may unlock secrets about our past, gain a deeper understanding of our present, and perhaps even catch a glimpse of the possibilities that lie ahead. The answers, when we finally find them, are sure to challenge our perceptions and redefine our place in the grand tapestry of life. To comprehend the enigma of Rh-negative blood, we must first grasp the basics of blood groups. Our blood, a complex mixture of cells and plasma, is categorized into different groups based on the presence or absence of specific proteins called antigens on the surface of red blood cells. These antigens, acting like cellular fingerprints, determine our blood type and play a crucial role in blood transfusions and immune responses. The Rh factor, named after the rhesus monkeys in which it was first discovered, is one such antigen. Denoted by the presence, plus or absence, of the D antigen, it determines whether an individual is Rh positive or Rh negative. For the vast majority, roughly 85% of the population, the Rh factor is present, inherited from their parents and passed down through generations. This prevalence suggests that the Rh factor offers a survival advantage, although its exact function remains a subject of ongoing research. Some scientists theorize that the Rh factor might play a role in cell membrane structure and integrity, while others speculate about its potential involvement in immune system regulation. Regardless of its precise function, the presence of the Rh factor is considered the norm, a testament to its long-standing presence in human evolution. However, the existence of a significant minority lacking this seemingly ubiquitous protein challenges this assumption, raising intriguing questions about the origins and implications of Rh-negative blood. The prevalence of the Rh factor in humans, mirroring its presence in other primates, suggests a shared evolutionary ancestry. If we subscribe to the theory of evolution, tracing our lineage back through millennia to common ancestors, the presence of the Rh factor makes perfect sense. It represents a genetic trait passed down through countless generations, a testament to our interconnectedness with the natural world. However, the existence of Rh-negative individuals disrupts this seemingly straightforward narrative. If the Rh factor is a fundamental component of our evolutionary heritage, then how do we explain the absence of this protein in a significant portion of the population? This discrepancy poses a challenge to traditional evolutionary models, prompting scientists and researchers to seek alternative explanations for the emergence of Rh-negative blood. Could it be that the Rh-negative blood type represents a more ancient lineage, a remnant of a time before the emergence of the Rh factor? Or is it a more recent development, a mutation that occurred spontaneously within the human genome? The answers to these questions remain elusive, shrouded in the mists of time and the complexities of genetic inheritance. The very existence of Rh-negative blood compels us to question our assumptions about human evolution, to delve deeper into the genetic archives of our species in search of answers. One prevailing theory suggests that the Rh-negative blood type arose from a genetic mutation, a sudden and random alteration in the DNA sequence responsible for producing the Rh factor. Mutations, the driving force behind evolution, introduce variations in genetic code, some beneficial, some detrimental, and many with no discernible effect. Proponents of this theory argue that the Rh-negative mutation, occurring sometime in the distant past, spread through specific populations due to a combination of factors, including geographic isolation, genetic drift, and potentially even natural selection. If the mutation offered a survival advantage in certain environments or conferred resistance to specific diseases, it could have become more prevalent in those populations over time. However, the exact nature and timing of this hypothetical mutation remain unclear. Some researchers speculate that it may have occurred relatively recently, within the last few thousand years, while others believe it could date back much further, potentially even predating the emergence of Homo sapiens. The lack of conclusive evidence makes it difficult to pinpoint the precise origin of the Rh-negative blood type, leaving the door open to alternative theories and interpretations. Understanding how Rh-negative blood is inherited provides further clues to its origins. Like other blood types, the Rh factor is determined by genes passed down from parents to offspring. Each parent contributes one of their two Rh genes, with the combination of these genes determining the child's Rh status. If both parents carry at least one Rh-negative gene, there is a chance their child will inherit two Rh-negative genes and therefore have Rh-negative blood. The probability increases if one or both parents are themselves Rh-negative. This pattern of inheritance suggests that the Rh-negative trait is recessive, meaning it only manifests when an individual inherits two copies of the Rh-negative gene. This recessive nature adds another layer of complexity to the puzzle of Rh-negative blood. If the Rh-negative trait is relatively rare, then how did it manage to persist and spread through generations? One possibility is that it offered a survival advantage in certain environments or provided protection against specific diseases, giving individuals with Rh-negative blood a better chance of survival and reproduction. The uneven distribution of Rh-negative blood across the globe offers tantalizing clues to its origins and historical spread. While it is found in trace amounts in most populations, certain regions and ethnic groups exhibit significantly higher concentrations, suggesting a shared ancestry or unique genetic events in their past. Mapping the prevalence of Rh-negative blood reveals fascinating patterns. The highest concentrations are found among the Basque people of the Pyrenees Mountains, straddling the border of Spain and France, and among Eastern Oriental Jewish populations scattered across Europe, the Middle East, and beyond. These groups, often geographically and culturally isolated, have preserved unique genetic markers, including the Rh-negative trait, passed down through generations. The geographical clustering of Rh-negative individuals suggests a common origin, a point source from which the trait spread outwards. Whether this origin lies in ancient migrations, genetic bottlenecks, or other factors remains a subject of debate. By studying the genetic makeup of these populations and comparing it to surrounding groups, researchers hope to unravel the mysteries of Rh-negative blood and its journey through time and space. Nestled in the rugged Pyrenees Mountains, the Basque people have long captivated anthropologists and geneticists alike. Their unique language, unrelated to any other known tongue, and their distinct cultural traditions, passed down through generations, hint at an ancient and isolated past. Adding to their mystique is the remarkably high prevalence of Rh-negative blood among the Basque population, with some estimates exceeding 30%, the highest concentration found anywhere in the world. This genetic peculiarity has fueled speculation that the Basques represent a remnant of an ancient European population, perhaps even predating the arrival of Indo-European groups thousands of years ago. Some theories propose that the Basque country served as a refuge during the last ice age, allowing them to preserve their unique genetic heritage while other populations migrated or intermingled. The Basque language, Euskara, further deepens the mystery. With no known linguistic relatives, it stands as an anomaly among European languages, its origins shrouded in the mists of time. Some linguists speculate that it may be a relic of a pre-Indo-European language family, spoken across Europe before the arrival of the Indo-European languages that dominate the continent today. The combination of their unique language, distinct cultural practices, and high prevalence of Rh-negative blood has led some to view the Basques as a living link to Europe's distant past, guardians of a genetic and cultural heritage that has all but vanished elsewhere. Eastern Oriental Jewish populations, dispersed across the globe yet bound by shared ancestry and religious traditions, also exhibit a higher than average incidence of Rh-negative blood. While the exact percentages vary among different Jewish communities, the prevalence is notably higher than in surrounding non-Jewish populations, suggesting a common genetic thread woven through their history. Several theories attempt to explain this phenomenon. One possibility is that the Rh-negative trait was present in the ancestral Jewish population that emerged in the Middle East thousands of years ago. As Jewish communities migrated and settled in different parts of the world, they carried this genetic marker with them, preserving it at higher rates due to their religious and cultural practices. which often encouraged marriage within the faith. Another theory proposes that the higher prevalence of Rh-negative blood among Eastern Jews may be linked to historical events, such as the Babylonian exile or other periods of persecution and forced migration. These events could have led to genetic bottlenecks, reducing population sizes and increasing the frequency of certain traits, including the Rh-negative blood type, within the surviving communities. The genetic history of Eastern Jews, marked by both continuity and upheaval, offers a compelling case study in the complex interplay of genetics, culture, and historical forces. The geographical distribution of Rh-negative blood, with its clusters and pockets of higher prevalence, points to the powerful role of migration and intermingling in shaping human genetic diversity. As populations moved across continents, interacting with other groups and adapting to new environments, they carried their genes with them, leaving behind traces of their journeys in the genetic makeup of modern populations. The Rh-negative trait, like other genetic markers, spread through these ancient migrations, its frequency influenced by factors such as founder effects, genetic drift, and potentially even natural selection. Founder effects occur when small groups break away from larger populations, carrying with them a limited gene pool that may not reflect the full diversity of the original group. Genetic drift, on the other hand, refers to random fluctuations in gene frequencies over time, particularly pronounced in small, isolated populations. Natural selection, the driving force behind evolution, may have also played a role in shaping the distribution of the Rh-negative trait. If this blood type offered a survival advantage in certain environments or conferred resistance to specific diseases, it could have become more prevalent in those populations over time. However, the exact nature of any potential selective pressures remains a subject of speculation. Unraveling the complex interplay of these forces, tracing the movement of genes across continents and through millennia, is crucial to understanding the global tapestry of human genetic diversity and the enduring enigma of Rh-negative blood. The exploration of Rh-negative blood extends beyond its genetic origins and geographical distribution, venturing into the realm of human phenotype, the observable characteristics and traits that make each individual unique. While science paints a complex picture of blood groups and their inheritance, anecdotal evidence and popular culture have woven an intriguing, albeit often unsubstantiated, narrative around Rh-negative individuals, attributing to them a range of unusual physical and psychological characteristics. These claims, ranging from the mundane to the extraordinary, have captivated the public imagination, fueling speculation and debate about the true nature of Rh-negative blood. Some attribute to these individuals higher than average IQs, increased psychic abilities, and even a heightened sensitivity to sunlight. Others describe physical characteristics like predominantly green or hazel eyes, a reddish tint to their hair, and an extra vertebra. While these claims lack rigorous scientific backing, their persistence in popular culture speaks to our fascination with the unknown and our desire to find meaning in the seemingly random patterns of human biology. It's essential to approach these claims with a healthy dose of skepticism, separating anecdotal observations from scientifically validated evidence. While some Rh-negative individuals may indeed possess certain traits, attributing these traits solely to their blood type risks falling into the trap of correlation versus causation. Human phenotype is a complex interplay of genetics, environment, and chance, and attributing specific traits to a single blood type oversimplifies a multifaceted phenomenon. One of the most persistent claims surrounding Rh-negative individuals is their supposed connection to higher intelligence and psychic abilities. Anecdotal accounts abound of individuals with Rh-negative blood exhibiting exceptional cognitive skills, heightened intuition, and even telepathic abilities. These claims, often amplified in popular culture and paranormal circles, have fueled a sense of mystique and intrigue around this blood type. However, scientific studies have yet to establish a causal link between Rh-negative blood and enhanced cognitive function or psychic abilities. While some studies have reported a slightly higher average IQ among Rh-negative individuals, These findings are often statistically insignificant or fail to account for other factors like socioeconomic background, education level, and cultural biases in IQ testing. Similarly, claims of psychic abilities, often subjective and difficult to measure, lack the rigorous scientific evidence required for validation. Another common claim links Rh-negative blood to physical characteristics like green or hazel eyes, reddish hair, and even an extra vertebra. While these traits may be more prevalent in certain populations with higher concentrations of Rh-negative blood, attributing them solely to blood type ignores the complex interplay of genes that determine human phenotype. Eye and hair color, for example, are determined by multiple genes, each with its own set of alleles and inheritance patterns. While certain combinations may be more common in specific populations, they are not exclusive to individuals with Rh-negative blood. Similarly, anatomical variations like an extra vertebra, while intriguing, are not unique to any particular blood type and can result from a range of genetic and developmental factors. Despite the lack of conclusive scientific evidence, the allure of the unknown continues to fuel fascination with Rh-negative blood. The human mind, wired for pattern recognition and meaning-making, seeks explanations for the unexplained, often filling in the gaps with speculation, mythology, and cultural narratives. The rarity of Rh-negative blood, particularly in certain populations, adds to its mystique. Throughout history, rare and unusual phenomena have often been attributed to supernatural forces, divine intervention, or even extraterrestrial influence. The Rh-negative blood type, shrouded in genetic mystery and often associated with ancient lineages and isolated communities, becomes fertile ground for such speculation. The internet age, with its rapid dissemination of information and its tendency to amplify both fact and fiction, has further fueled the fascination with Rh-negative blood. Online forums and social media groups dedicated to this blood type abound, with members sharing personal anecdotes, exploring alternative theories, and perpetuating myths and misconceptions. While these online communities can foster a sense of belonging and shared identity, they can also contribute to the spread of misinformation and the blurring of lines between scientific inquiry and pseudoscientific speculation. The enigma of Rh-negative blood ultimately boils down to a fundamental question. Does it represent a distinct genetic lineage, a remnant of ancient human populations, or even extraterrestrial influence, as some theories propose? Or is it simply a statistical anomaly, a random mutation that has persisted and spread through specific populations due to a combination of genetic drift, founder effects, and potentially even natural selection? The answer, like many things in science, is likely more nuanced than a simple either-or proposition. While the scientific community generally accepts that the Rh-negative blood type arose from a mutation, the exact nature and timing of this mutation remain unclear. Further research into the genetic makeup of populations with high concentrations of Rh-negative blood, combined with advances in ancient DNA analysis and a deeper understanding of human evolutionary history, may shed light on this enduring mystery. Regardless of its origins, the Rh-negative blood type serves as a reminder of the incredible diversity and complexity of human biology. It challenges us to question our assumptions, to explore the boundaries of scientific knowledge, and to embrace the mysteries that continue to captivate our imaginations. The quest for artificial blood has long captivated the imaginations of scientists and medical professionals, promising a revolutionary solution to blood shortages and transfusion complications. While significant strides have been made in replicating the oxygen-carrying capacity of red blood cells, recreating the complex interplay of antigens, proteins, and cellular interactions that define our blood types, remains a formidable challenge. This challenge is particularly pronounced when it comes to Rh-negative blood, presenting a unique set of obstacles that have thus far stymied scientific endeavors. The absence of the Rh factor, a seemingly minor variation in blood composition, has profound implications for blood transfusions. Rh-negative individuals can only receive blood from other Rh-negative donors, as exposure to Rh-positive blood triggers a potentially life-threatening immune response. This incompatibility, a consequence of our immune system's remarkable ability to recognize and target foreign invaders, creates a constant demand for Rh-negative blood, a demand that often outstrips supply. The creation of artificial Rh-negative blood, capable of seamlessly integrating into the circulatory system without triggering an immune response, would be a game changer for millions worldwide. It would alleviate the pressure on blood banks, reduce the risk of transfusion reactions, and potentially even pave the way for personalized blood products tailored to individual patients. However, despite decades of research and countless scientific breakthroughs, the dream of readily available, universally compatible artificial blood, particularly for Rh-negative individuals, remains elusive. One of the most intriguing aspects of Rh-negative blood, particularly from a scientific standpoint, is its resistance to cloning. While the protein responsible for the Rh factor in positive blood can be replicated in laboratory settings, attempts to clone the absence of this protein to create Rh-negative blood from scratch have consistently met with failure. This peculiar characteristic has fueled speculation and debate, blurring the lines between scientific inquiry and more unconventional interpretations. Some researchers speculate that the inability to clone Rh-negative blood may stem from the complex interplay of genes and regulatory mechanisms involved in blood type determination. The absence of the Rh factor is not simply a matter of removing a single protein. It may involve a cascade of genetic and cellular interactions that have yet to be fully deciphered. Others, venturing into more speculative territory, propose that the resistance to cloning could be a sign of something more profound, a hint that Rh-negative blood possesses unique properties or origins that defy conventional scientific understanding. While these alternative interpretations captivate the imagination, it's essential to approach them with a healthy dose of skepticism. The scientific community, while acknowledging the challenges in cloning Rh-negative blood, generally attributes them to the complexities of blood cell development and the intricacies of genetic regulation. Further research into the molecular mechanisms underlying blood type determination, coupled with advances in cloning technologies, may eventually overcome these obstacles, paving the way for the creation of artificial Rh-negative blood. The quest to understand and potentially manipulate Rh-negative blood, while driven by noble goals of improving healthcare and saving lives, also raises a host of ethical considerations that warrant careful examination. As we delve deeper into the genetic underpinnings of blood types and explore the possibilities of artificial blood creation, we must grapple with the potential consequences and ensure that scientific advancements align with ethical principles and respect for human diversity. One ethical concern revolves around the potential for genetic discrimination. If Rh-negative blood is perceived as unique, valuable, or even linked to certain desirable traits, as some theories suggest, it could lead to the stigmatization of individuals with other blood types, or even attempts to selectively breed for or against the Rh-negative trait. Such practices would raise serious ethical concerns, echoing historical instances of eugenics and discrimination based on perceived genetic superiority. Another ethical consideration pertains to the allocation of resources and access to potentially life-saving treatments. If artificial Rh-negative blood becomes a reality, it's crucial to ensure equitable access, regardless of socioeconomic status or geographic location. The benefits of this scientific breakthrough should be available to all who need them, not just those who can afford it or reside in affluent regions. The pursuit of scientific knowledge and medical advancements should always be guided by principles of justice, fairness, and the well-being of all members of the human family. The journey into the world of Rh-negative blood has taken us through winding paths of genetics, history, anthropology, and even touched upon the fringes of speculation and myth. We've delved into the intricate workings of our blood, exploring the significance of the Rh factor and the perplexing absence thereof in a small yet significant portion of the population. We've traversed continents, tracing the geographical distribution of this unique blood type, encountering ancient cultures like the Basques and Eastern Jews, who, with their unusually high concentrations of Rh-negative individuals, offer tantalizing clues to a shared past shrouded in mystery. We've grappled with scientific theories, attempting to explain the origins of Rh-negative blood, weighing the evidence for genetic mutations, ancient lineages, and the ongoing debate between conventional explanations and more unconventional interpretations. We've ventured into the realm of human phenotype, exploring the claims of unique physical and psychological characteristics associated with Rh-negative blood, separating anecdotal observations from scientifically validated evidence, acknowledging the allure of the unknown, while upholding the importance of rigorous scientific inquiry. And yet, despite our best efforts, the enigma of Rh-negative blood persists. It stands as a testament to the vastness of what we don't know, a reminder that even in the age of advanced scientific knowledge, some mysteries endure, challenging us to expand our understanding of human biology, history, and the very essence of what it means to be human. As we stand at the precipice of new scientific frontiers, the enigma of Rh-negative blood beckons us forward, inviting continued exploration and promising potential breakthroughs that could reshape our understanding of human origins and the intricacies of our genetic makeup. Advances in genomic sequencing, ancient DNA analysis, and our growing knowledge of human migration patterns hold the key to unlocking the secrets hidden within this unique blood type. The quest for artificial blood, driven by the noble goal of saving lives and alleviating suffering, will undoubtedly continue, fueled by the hope of one day overcoming the challenges posed by Rh incompatibility. The scientific community, armed with ever-evolving tools and technologies, will continue to push the boundaries of what's possible, seeking innovative solutions to replicate the complexity of human blood and create universally compatible blood products. Yet, as we venture further into this uncharted territory, we must proceed with caution, guided not only by the pursuit of knowledge, but also by a deep respect for human diversity and the ethical implications of our actions. The potential for genetic discrimination, the importance of equitable access to life-saving treatments, and the need to safeguard against unintended consequences must guide our steps as we navigate the complex landscape of Rh-negative blood research. The enigma of Rh-negative blood is more than just a scientific puzzle. It's a reflection of our collective curiosity, our innate desire to understand the world around us and our place within it. It's a story that spans millennia, connecting us to ancient ancestors, lost civilizations, and the enduring mysteries of human evolution. As we move forward, let us embrace the spirit of open-minded inquiry, fostering collaboration between scientists, historians, anthropologists, and curious minds from all walks of life. Let us encourage rigorous research while remaining open to alternative perspectives, always grounding our explorations in ethical considerations and a deep respect for the diversity of human experience. The enigma of Rh-negative blood, with its tantalizing clues and unanswered questions, serves as a powerful reminder that the pursuit of knowledge is a journey without a final destination. It's a continuous exploration, a quest driven by curiosity, fueled by wonder and guided by the enduring human spirit of discovery.